Set 34 clinical trial data, technical question and answers. Answers are highlighted in green. Number 1. Missing visit data. Scenario. In the SDTM VS Vital Signs dataset, some subjects have missing visit numbers but have visit dates available. So the question here is, how would you assign the correct visit num and visit variables using the available data? Now you can use the SV subject visits dataset and match visit dates using date windows to derive correct visit and visit number. Reason is, visit information is usually derived from protocol defined visit schedules and visit date ranges. Number 2, duplicate records. Scenario. During validation, you find duplicate records for the same subject, visit and test in the LV domain, which is the laboratory. Question. How would you identify and resolve duplicates? You can identify duplicates using proc sort data is equal to, this is the name of the data set. You can use the no duplicate option here by using variables, primary key variables, which is unique subject ID, the LB test code and the LB date character. Then you can also use the numeric one also, and then you can verify whether the duplicates are true duplicates or repeated assessments, because duplicates must be clinically reviewed before deletion, because repeated measurements may be valid. Number 3, partial date problem scenario. In the AE domain, adverse event start date is recorded as 2025-05 month only. Question. How would you derive AE start date for analysis datasets? You can apply protocol or SAP defined imputation rules if it is mentioned in protocol and SAP, and you can simply assign 01. In some studies they use 15 or they use month end date, which is 30 or 31st. So it depends on what rules it is satisfied in protocol and SAP. Reason is, partial dates are imputed conservatively to avoid incorrect treatment emergent classification. Number 4, adverse event before treatment scenario. An adverse event start date is before the first dose date. Question. Should this AE be considered treatment emergent? No. The AE is generally not treatment emergent if AE start date is before first dose date. Explanation here is, treatment emergent AEs are events that start or worsen after treatment initiation. Number 5, lab units conversion scenario. Lab values come from multiple countries with different units. Say for example, I have a lab test hemoglobin. The values are 13 and 130. So here the units are different, which is gram per deciliter, and here it is gram per liter. So the question here is, how would you standardize lab values? So you can convert all lab values into standard units using conversion factors and populate this lab standard result numeric variable and the units variable. So the explanation here is, standardized lab units ensure consistent analysis across countries and sites. Number 6, population flag derivation scenario. You need to create safety population SAFFL. Question. What rule would you use? Answer here is, you can set SAFFL is equal to Y for subjects who received at least one dose in EX dataset. Explanation here is, safety population includes all treated subjects. Number 7, visit window problem scenario. A subject attended visit 3 2 days earlier than planned. Question. How would you determine if this visit belongs to visit 2 or visit 3? Answer here is, assign visit based on protocol defined visit window. Say for example, day 28 plus or minus 3 days assigned to visit 3. Explanation here is, visit windows prevent incorrect visit assignment due to scheduling deviations. Number 8, death date before last visit scenario. A subject has death date earlier than last recorded visit date. Question. How would you investigate this? Now you can raise a data query and verify source data because chronology is inconsistent. Death cannot logically occur before a later recorded visit. Number 9. Inconsistent sex information. Scenario. Sex in DM dataset is equal to male, but pregnancy test exists in LB dataset. How would you handle this issue? Investigate source data and raise query for potential demographic coding error. Pregnancy test for male subjects indicate possible data inconsistency. Number 10. Subject appears in AE but not in DM. A subject exists in AE dataset but not in DM dataset. What steps would you take? Verify unique subject ID, screen failure status, and data transfer completeness. All analysis subjects should exist in the DM domain. Number 11. Randomization mismatch. Subject randomized to drug A but EX dataset shows drug B. What would you check? You can check the randomization dataset, you can check the EX dataset, IWRS, IRT records, you can investigate dispensing or mapping errors. Treatment assignment mismatches may indicate serious operational issues. Number 12. Visit date before informed consent. Visit date recorded before consent date. Question. What does this indicate? You can raise protocol deviation query because study procedures before consent are usually not allowed. Subjects must provide consent before participating in trial activities. Number 13. Baseline flag derivation. How do you derive baseline flag ABL-FL when multiple records exist before dosing? You can assign ABL-FL is equal to yes to the latest non-missing assessment before first dose, date and time. Baseline represents the last assessment before treatment exposure. Number 14. Lab value outlier. Potassium value recorded as 45 millimole per liter. What steps would you take? You can check the unit conversion, you can check the decimal point errors, clinical possibility, then the source data verification. Extreme lab values would indicate data entry or unit conversion issues. Number 15. Time point missing. PK samples have time points missing but collection times available. How would you derive time points? You can derive time points using sample collection date time, dose, date time, protocol, schedule. PK time points are critical for accurate pharmacokinetic analysis. Number 16. Treatment emergent adverse events TEAE. How would you identify treatment emergent adverse events TEAE using AE and EX datasets? Now a subject is considered to have treatment emergent AEs if AE start date is greater than or equal to first dose date. You can also apply additional SAP versioning rules that have been mentioned and confirmed with sponsor. Treatment emergent AEs identifies adverse events associated with treatment exposure. Number 17. Data traceability. Why is traceability important in ADAM? To ensure every analysis value can be traced back to SDTM and /raw data. Traceability is required for regulatory compliance and audit readiness. Number 18. Control terminology. Why are control terminologies important? They standardize data across studies and regulatory submissions. Control terminology improves consistency and interpretability. Many thanks for watching my video and happy learning. Please like, share, subscribe, leave a feedback comment. Many thanks for your support and having great connection.